My name is Bianca Vora, and I'm a graduate student in the Giacomini Lab at the University of California, San Francisco, and today I'll be talking about my project, which utilizes a multifaceted approach to understand transporter-mediated drug-nutrient interactions. Vitamin B1, or thiamine, is a water-soluble vitamin which is obtained exclusively from our diet. Thiamine transporter 2, or THTR2, is the primary absorptive thiamine transporter in the intestine. Recent data suggests that commonly used prescription drugs, such as trimethoprim, are predicted to inhibit THTR2 at clinically relevant intestinal concentrations. The goal of this study was to determine the effects of the antibacterial drug trimethoprim on thiamine concentrations in healthy volunteers by employing a multifaceted approach using in vitro, clinical trial, and real-world data. 6 subjects were administered a single oral dose of thiamine or trimethoprim plus thiamine, followed by intensive sampling for up to 24 hours post-dose. The effect of trimethoprim on transporter-mediated thiamine uptake was investigated in stably transfected HEK cells overexpressing the transporter. Electronic health record data were obtained from the UCSF Research Data Browser. Results in these 6 volunteers showed that thiamine plasma concentrations increased following co-administration with trimethoprim. The maximum concentration achieved, or Cmax, in area under the curve, or AUC, was significantly higher when thiamine was co-administered with trimethoprim. In vitro data demonstrated that trimethoprim is a potent inhibitor of OCT1, suggesting that trimethoprim may inhibit OCT1-mediated thiamine uptake into the liver in vivo, thus reducing metabolism of thiamine to its metabolites and elevating its plasma levels. From the EHR data, we observed that HIV patients prescribed trimethoprim had significantly higher triglyceride, LDL cholesterol, and total cholesterol levels, a known consequence of OCT1 inhibition. Our study highlights that co-administration of trimethoprim with thiamine increased rather than decreased plasma levels of thiamine, potentially through inhibition of OCT1-mediated hepatic uptake. Trimethoprim should be considered for patients who may be at risk for thiamine deficiency. However, alternative antibiotics should be considered for patients who may be predisposed to increased lipid levels and are chronically taking trimethoprim. Using multiple levels of evidence, the study suggests that although OCT1 is an established target for drug-drug interactions, OCT1 may be a target for drug-nutrient interactions as well. With that, I would like to acknowledge and thank all the subjects for their participation, and thank you, ARCS, for allowing me this opportunity to present my works. Thank you.